I am Patricia Beaston, a clinical reviewer in the Office of Tissue and Advanced Therapeutics. Today, I will give a brief introduction to clinical considerations for functional studies of pig kidneys that will be used for transplantation. With improvements in surgical techniques and tools, donor-recipient matching, and immunosuppressive regimens, the success of transplantation can exceed 90% at 1 year, and 10-year survival has surpassed 50%. The success of kidney transplant is greater than that for liver transplant, which is greater than that for heart transplant, with living donor transplant more successful than cadaveric donor transplants. While these are life-saving and life-improving strides, there is a shortage of donors, living or deceased, compared to the number of patients on waiting lists, and some potential recipients have characteristics that make achieving a match near impossible. To address this imbalance between the need for transplantation and the availability of donors, the use of organs from other species has been considered for more than a century, with tissues and cells being investigated within the more recent past. Please note that allogenic organ transplant is regulated under the Health Resources and Services Administration, or HRSA, whereas xenotransplantation products are regulated as a biologic under Part 610 of the Code of Federal Regulations. We must remember that the purpose of renal transplant is to provide replacement function for kidneys that are no longer able to support life in patients. Therefore, it is important to consider whether the product obtained from the source animal is sufficient to approximate the physiology of the human kidney that it is meant to replace. Surgical techniques for kidney transplantation are well established. However, there are no data to determine the appropriate criteria for organ selection, such as the age of the source pig or the size of the organ. The clinical review starts with input provided by the chemistry and manufacturer controls, or frequently referred to as CMC, and pharmacotoxicology PT reviewers, as this information forms the basis for the evaluation of the safety mitigations contained within the proposed clinical protocol. The CMC reviewer determines that the kidney obtained from the source animal meets the requirements for transplantation. A major risk for xenotransplantation is the risk of implanting an infected organ, which may fail in an immune-compromised recipient and/or may result in zoonotic infections spread from one animal species to another. So I was talking about the zoonotic infections and the spread from one species to another, in this case, pig to human. However, the discussion of zoonosis is beyond the scope of today's brief presentation. The pharmacology toxicology reviewer considers whether the animal model is appropriate for the clinical condition or disease. These considerations include, but are not limited to, the route of administration, which should mimic the proposed clinical route as much as possible, and include the surgical approach, delivery devices, concomitant medications, and immunosuppression regimens that would be the same or similar to those as proposed for the clinical study. While immunosuppressive regimens for allogenic transplants are well established, immunosuppressive regimens that are appropriate for the xenokidney are not well established. The pharmacology toxicology evaluation of immunosuppressive regimens for xenotransplantation in non-human primates is limited because the commonly used drugs may not be as effective or well tolerated in the non-human primate. This may also limit the ability to demonstrate prolonged function of the transplanted organ. To assess the proposed clinical studies, the clinical team considers data gathered from the preclinical study endpoints for safety and organ function. I will introduce two of the major potential safety issues that would be considered in the review of the proposed clinical study. In general, if the transplanted organ, tissues, or cells cannot meet or approximate the replacement of the human organ, this mismatch can pose a risk to the recipient. Allogenic kidney transplant has the expectation that the donor kidney will provide replacement therapy. The move to xenotransplantation requires consideration of the kidney's functions and the need to explore whether the xenokidney can provide replacement for all of these functions, and if not, can the risk of this physiologic mismatch be mitigated? In addition to waste removal, the kidney regulates electrolytes and is a complex an organ that produces, converts, and responds to hormones. The actions of these hormones are not always conserved across species. I will describe a few examples of these complex functions. We will start with fluid balance, blood pressure, and electrolyte balance. Potassium and phosphate wasting has been reported in pig to cynomolgus monkey bilateral nephrectomy model. Free water wasting has been reported in the non-human primate model and raises concern for a mismatch for response to vasopressin. In sodium regulation, salt excretion is influenced by several natriuretic peptides which act on the kidney, and salt sparing is achieved through the renal sympathetic nervous system and the renin-angiotensin-aldosterone axis. We know that porcine renin does not cleave human angiotensinogen. The vitamin D parathyroid or PTH axis is critical to maintaining calcium and phosphate levels within the appropriate physiologic range. In response to PTH, the kidney is the site of 1-alpha hydroxylation of 25 vitamin D to produce the active form of vitamin D, which promotes intestinal absorption of calcium and phosphate. PTH also promotes tubular reabsorption of calcium while inhibiting phosphate reabsorption. The amino acid sequence for PTH is not conserved between humans and pigs, and the response of the pig kidney to human PTH has not been described. Porcine erythropoietin is only 80% homologous to the non-human primate erythropoietin and does not support non-human primate erythropoiesis. Similarly, porcine erythropoietin does not support human erythropoiesis. While not unique to the kidney, it should be noted that pigs and primates have a mismatch in the coagulation cascade. This mismatch can increase the risk of thrombus formation and requires consideration during the transplant and post-transplant periods. We must also consider the pharmacokinetics and pharmacodynamics of drugs that will be used in the peri-transplant period to provide immunosuppression and to manage the recipient's other comorbidities or complications that may occur from the transplantation procedure or immunosuppression. There are drugs such as the SGLT2 inhibitors for treatment of diabetes that act on the kidney. It is important to understand whether the xenokidney and the human kidney have similar responses to these drugs. In addition, the xenokidney and the human kidney may be different in how they metabolize some drugs. This difference could result in underdosing, leading to ineffective therapy, or overdosing, leading to possible toxicity. Such differences in metabolism could be most critical for the drugs that have a narrow therapeutic index. Additionally, some drugs could be toxic to organs. It is important that the drugs used in the post-transplant period are not toxic to the transplanted organ. In summary, the FDA considers the potential benefits and the potential risks at all stages of clinical development. The hope for benefit is for the transplanted organ to provide the intended physiologic and functional replacement. However, with this benefit comes many risks, both known and unknown. Risks from the route of administration include risks associated with implantation procedures, such as bleeding and infection, and risks associated with the site of implantation based on the organ to be transplanted. Novel immunosuppressive regimens and risk from infection, including zoonosis, present additional risks that remain to be characterized. Today, I've presented a brief discussion of considerations for physiologic mismatch in the case of kidney transplantation and considerations for clinical pharmacology. For recipient safety, it's important to consider the requirements for the transplanted organ, in our example today, the pig kidney, to provide replacement therapy. The clinical protocol should identify the risks associated with the proposed treatment and provide a specific plan to mitigate these risks. Such a plan should consider subject eligibility criteria, treatment plans, safety monitoring, and management of physiologic mismatch. For those who are interested in additional regulatory information, this slide contains links to guidance documents and webinars that can provide valuable information on the CMC and PT requirements, as well as those for IND submission and how to request meetings with the FDA. Thank you for your attention.
Thanks so much, Dr. Beaston. Any questions from our audience for Dr. Beaston?
Hello, this is Dr. Wieringa from the Netherlands. Big compliments to FDA for the way you're reaching out for innovation that is super duper. And also the way you explain now the plans on how to proceed on this. Would an IDE status also be possible for such a product, an investigational device exemption, or is this something which does not exist for the biological things?
Doug?
Well, certainly, if somebody wants to do a clinical study for a device, especially for a device that's a significant risk study, then it would require an IDE if you want to do this study in the United States. And we certainly would convey that we encourage all innovators, whether they be within the U.S. or outside the U.S., to conduct some studies in the United States, especially if you intend to market the devices in the United States, because there may be some population-specific issues which can only be addressed by doing a study that includes U.S. patients. We see many IDEs for various types of devices for various indications for which an IDE is submitted. Now, very often, if not always, a company will come into us prior to the IDE submission phase with what we call pre-submission or a Q submission if they want to discuss certain aspects of their preclinical data and or their clinical data. And we encourage the Q submission because it allows you to get some information, not just simply on the clinical study, but also on how you want to design and organize your preclinical studies, your bench studies. So we encourage you to come into us because doing that beforehand, before you start your studies, while you're even in the early phases of device development, might actually cut off some time and requirements as you go further along. You don't want to do it, go far along in device development and find that there is a problem that you have to go back and redesign. And I'll defer to Dr. Beaston on some of the requirements to do a clinical study for biologics. But as I said, we encourage you to come in early, send us an email and say you want to discuss something. We'll walk you through the process and we'll be there the entire way along with you. We'll outline, as Dr. Beaston just did for these Xenotransplantation studies, we'll outline what the requirements are from literally throughout your entire study design for preclinical and the clinical phase.
That's huge. I'm very impressed by the super attitude of FDA and reaching out to innovation and to the patients that they're serving. Really, that's great.